So this okay, I'm trying to this one the screen. Okay, all right. And then we have the tumor suppression genes, right? So these tumor suppressor genes are genes that encode for the proteins that participate especially in the cell cycle, all right? So tumor suppressor gene is also called anti-oncogene, all right? This tumor suppressor gene regulates cell during cell division. Okay, so its effect is against the oncogene or anti-oncogene. All right. So tumor suppressor genes also can be multiple. They can be either receptors for different growth factors or enzymes that control DNA repair, and loss of expression loss of expression of tumor suppressor genes also carries the risk of malignancy. On the other hand, besides the oncogenes and tumor suppressor genes, we have the epigenetic alterations, all right? So epigenetic alterations actually alter the gene, but it doesn't change the structure of the DNA or it doesn't change the nucleotide sequence, but functionally it affects the working of the DNA or the genome, all right? So the latest terminology or definition or description of epigenetic alteration is a structural adaptation of chromosomal regions so as to register signal or perpetuate altered activity states. In other words, epigenetic alteration will affect the function of the genes without affecting the structure of the nucleotide sequence or the genes. I will illustrate this in a few slides after this. Okay, next slide. Okay, this is a picture to show the oncogenes and the proto-oncogenes. We have known oncogene is the gene that has potential cancer, and then these genes are mutated in tumors. All right, most normal cells will undergo programmed cell death or apoptosis. All right. When the critical functions are altered or malfunctioning, activated oncogenes can cause these cells to be designated for apoptosis. The cell becoming, there's a control over the proliferation. Once the oncogene is proto-oncogene is mutated, we call oncogene, and this will cause proliferation of the cells. The cells will not die. All right, and we know that after a certain period of time, the cells will die. But with mutation, the cells will not die, will proliferate. Okay, so this will cause accumulation of cells and abnormal cells in the organs that are affected. For example, in the bone marrow or in the other organs. Okay, so that's the function of oncogenes. Next slide, please. Okay, this picture shows the various types of oncogenes, okay, proto-oncogenes and oncogenes, okay. We have at least 5 groups of oncogenes, all right. For example, growth factors, and then growth factor receptors, also protein kinase, and also transcription factors. So in normal situation, the proto-oncogenes are exerting its function, and with mutation, all these normal mechanisms are suppressed. And the net result, one of the net result or outcome is there is no apoptosis and the cells will survive and proliferate. So that is the concept of oncogene in a nutshell. So the next slide, please. Okay, what about tumor suppressor genes? As mentioned earlier, tumor suppressor genes are anti-oncogene. In a normal situation, the tumor suppressor genes act like a brake pedal, all right? So in a normal situation, the example here is tumor suppressor gene proteins. You know, it acts on the signaling enzymes, also acts as receptor and transcription factors, all right? So it is involved in the cell cycle. So with mutation in the tumor suppressor gene, if we compare to the concept of the second diagram here, you know, if we look at the cell cycle, it's the cell cycle here is like analogy to a traffic light. All right. So the tumor suppressor gene, you know, when there is a mutation in the tumor suppressor gene, there is no brake in the synthesis of cells or production of cells. So again, the end result will be proliferation of cells. So there's no break in the synthesis in the cell cycle. And loss of expression of these genes is associated with high risk of developing a malignancy. Next slide, please. Okay, these are examples of tumor suppressor genes, which are RB gene or retinoblastoma gene, P53, all right? Also in B-cell lymphoma and neurofibromatosis gene. Okay, next slide, please. Okay, a little bit about the retinoblastoma gene. Is the first tumor suppressor gene identified, where studies on retinoblastoma was done, and this gene was discovered in 1986, where the retinoblastoma gene is either lost or mutated in patients with retinoblastoma. In experiments using gene transfer, where there is introduction of normal retinoblastoma gene into the cells. So when the normal gene is introduced into the retinoblastoma cells, the tumorigenicity or the malignancy potential of the cells are cancelled. In other words, the abnormal retinoblastoma gene will be replaced by the normal retinoblastoma gene. So this indicates the activity of this gene as a tumor suppressor. All right. So this shows the condition of the eye where the tumor has affected at the posterior region of the eyes of the patient. All right. Next slide, please. Okay, what about epigenetics alterations? So we'll go in detail. What is epigenetic alteration? So epigenetic alteration, as we have known earlier, there is a change in the structure of DNA, but without changing the coding sequence. So this can occur either when the methyl groups are added or removed from the DNA. Also, secondly, when changes are made to histones, where the histones bind to the DNA in the chromosomes. And most important epigenetic alteration that have been described are DNA methylation, histone modification, and also microRNA regulation. All right. So here, there is a chemical structure added to the gene, but it doesn't affect the general structure of the DNA. But in other words, it only affects the function of that DNA or the function of that genome. Okay. Next slide, please. Okay, so these are situations where there is a genetic alteration. All right, what are the mechanisms that can induce epigenetic alteration? As the slide shows here, epigenetic alterations can occur during development as well, for example, in utero, but also in childhood. And then things like environmental chemicals, drugs, pharmaceuticals, aging, and diet also can affect the epigenetics or the function of the DNA. Okay, so the processes, the mechanism, as we have seen earlier, is either DNA methylation or histones that are affected, histones, the proteins that are around which DNA can wind or coil. Okay, where it's called histone modification, and the most important is the function of that gene is affected. And from the slide here, we can see that the endpoints here is either it can cause cancer, autoimmune disease, mental disorders, or diabetes. So these are examples of epigenetic alterations that can be caused either of these mechanisms. Okay, next slide, please. Okay, so this is the summary of how epigenetic alterations can affect the function of the gene and can cause diseases. Okay, so we have environmental factors such as smoking, socioeconomic status as well, diet, air quality. So this will give rise to epigenetic modification or alteration via these three processes, either DNA methylation, histone modification, or non-coding RNAs, and this will cause alteration in the gene expression through the different mechanisms. For example, imprinting and development can cause disorders, diseases, and also cancer as well. So that's next slide, please. Okay, this is again a summary of how epigenetic alteration can affect us, so through aging, lifestyle, diet, and also not forgetting the gut microbiome. All right, this can initiate molecular events and genetic alterations. All right, so these epigenetic alterations, it takes many years to develop. It doesn't develop within a short time. So it also undergoes different processes from the normal to benign neoplasia, and eventually it will develop into cancer, all right? Which take about 15 to 20 years for cancer to develop. Next slide, please. Okay, this is actually from a landmark paper by researchers from California University, where they have identified or initiated algorithm which identifies previously undetected cancer driver genes. Okay, through a new algorithm that they have developed, they discovered that there is interplay between oncogenes, tumor suppressor genes, and also epigenetic alterations that will lead or make a person progress to have cancer. And this is actually not a single event. It can be a multiple or two-step event. Okay, so here we see that oncogene are mutated. If they become mutated, there is increased proliferation, and then the tumor suppressor gene is mutated as well. So there is also enhanced cell cycle or production of cells in the body, and at the same time the epigenetic alteration is there. All right, so all these three mechanisms, oncogene, tumor suppressor gene mutation, and also epigenetic changes or alterations actually give rise to hematological malignancies. All right, so this landmark paper actually is very important because during the process of predicting the algorithm, they also have discovered new epigenetic genes as well, which are important in the development of cancer. Next slide, please. Okay. This is another slide to show clonal hematopoiesis in relation to selective pressures and secondary hematological diseases. All right. So here we have, just a moment. Okay, here we have in green, in orange, the factors that can cause pressure, selective pressure on somatic variants, for example, chemotherapy, aging, exogenous stress, for example, smoking. All right. So this will cause, stresses such as chemotherapy, aging, and smoking, or also diet for that matter, can cause selective pressure on the somatic variants, where they will eventually develop into acute myeloid leukemia. All right. And immune selection is also thought to be one of the causes for development, progression from, normal cells to MDS or AML. And this slide also shows the percentage of blasts that are found in the different stages of development of cancer from MDS to AML, right? So this paper actually tells us, there is a clonal hematopoiesis based on what are the pressures and the secondary hematological diseases that make a person develop cancer. All right. Next slide, please. Okay, so having said all that, having looked at the molecular basis underlying the hematological malignancies and hematological disorders, we know that hematological disorders are a very diverse group of disorders, which can either be malignant, all right, and then it can also affect the red blood cell, for example, thalassemias and hemoglobinopathies, red cell membrane disorders, also red cell enzymopathy such as G6PD deficiency or pyruvate kinase deficiency. Okay, hematological disorders can also include bleeding disorders and thrombosis such as hemophilia A and B, von Willebrand disease, and also protein C and protein S deficiency. So there is a diverse group of hematological disorders. Next slide, please. Okay. All right, this is the normal workflow for lab diagnosis when we suspect a patient to have acute leukemia. All right, first we will do a full blood count to look at the morphology of the cells. We are very familiar with that, okay? And we also look at the counts, and we also, if we suspect the patient to have some malignancy, for example, AML, ALL, we will proceed to bone marrow aspirate and trophine biopsy. So this is what happens in a primary hospital. All right. So once the diagnosis of acute leukemia is made, for example, from all these basic investigations, usually the patients will be referred to specialized or hospitals that focus on treating hematological malignancies. All right. So in this referral hospital, they will proceed with immunophenotyping, cytogenetics, and molecular studies. So this is what usually happens in Malaysia. Sometimes some hospitals may also have immunophenotyping facilities. All right. But for further testing, for example, cytogenetic and molecular studies, we have to refer the patients to other treating hospitals that treat only hematological malignancies. All right, next slide, please. Okay, so if for immunophenotyping, or we call Flowcytometry, so this is one of the tests that we use. So this is a laser-based technology and is able to identify and quantify cell populations. Usually normal cells will have their own characteristic profile, and in malignant cells, they express abnormal or aberrant phenotype that may be useful for detection of the malignancies. All right, so this is an example of PET scan tool. I saw the video just now. So you also have PET scan tool over there in the hospital, right? So next slide, please. Okay, so in flow cytometry, the antibodies are labeled with different fluorochromes. Okay, and these fluorochromes recognize the pattern and intensity of the expression of the different antigens on the surface of the normal and leukemic cells. The cells are then put in suspension and manipulated into a stream of fluid. All right. So in this process, the cells are lined as single cells, which are interrogated by electronic detection system. Next slide, please. So this is a typical results from a patient with ALL T-cell lineage. The blood cells here express positive for CD3, TDT, CD34, CD7, and CD2. So these are the results from the flow cytometry or immunophenotyping results. All right. So sometimes immunophenotyping can give us the diagnosis of the patient, but you know, besides diagnosis, you know, sometimes we also can gather the prognosis of the patients as well. Okay, next slide, please. Okay, next we go to cytogenetics. This is a conventional cytogenetic, which is a classical karyotyping of chromosomes. The resolution here is very low. We can only detect between 3 to 5 megabyte, and however, it remains the most comprehensive method for assessing chromosome abnormalities, especially numeric and structural chromosome aberration. All right. So the slide here shows Philadelphia chromosome, where there is translocation of chromosome 9 and 22. So cytogenetics is essential for disease classification. Not only that, is important also for prognostic assessment and treatment decisions. Next slide, please. Okay, the cytogenetics technique, you know, is a bit lengthy. All right. First, we have to culture the cells, and then we have to harvest. Culture the cells may take, you know, at least overnight or 16 hours. All right. And then we need to harvest, and also eventually the slide preparation and staining. Okay. So what I have highlighted in blue is you know, a shortened way of doing cytogenetics or chromosomal karyotyping, where we use high resolution banding technique using methotrexate cell synchronization. All right, so this will shorten the process to, you know, the culture process to only you know, between it will shorten the culture process. All right, next slide, please. So this shows the conventional cytogenetics method. So we screen the metaphases under inverted microscope. Next slide, please. And we analyze the metaphase spread. Next slide, please. And these subsequent photos shows the lab activities of doing karyotyping. All right. First, we receive requests from hospitals, the bone marrow samples, or sometimes we get blood samples as well. And then we process the bone marrow samples with chromosome spreading. Next slide, please. And then staining, followed by slides ready for analysis and loading the slides into automated scanner and chromosome analysis and karyotyping. So these are the involved lab activities. Next slide, please. So this is an example of a cytogenetic platform, which is available in some hospitals in Malaysia. So this is used for image capturing and fish scoring. So it needs a very experienced staff to do all this, analysis and subsequently capturing as well. Next slide, please. So, looking at the, quite tedious way of analyzing the chromosomes, karyotyping still remains important in diagnosis and sometimes prognosis of the patient, all right? So the advantages of using cytogenetics is because it's able to detect additional cytogenetic abnormalities in the chromosome which cannot be detected by locus-specific techniques, for example, FISH or PCR. Of course, it has disadvantages. It cannot detect small deletion or few kilobase pairs length and cryptic translocation as well. It is laborious, as you can see just now. And also there's technical issues associated with cytogenetics, which resulted in increased use of molecular cytogenetics techniques such as FISH. However, it still remains the gold standard for most numerical and large structural chromosome abnormalities. Next slide, please. All right. Then we have this molecular techniques, which is FISH, fluorescent in situ hybridization, array CGH, polymerase chain reaction, which include reverse transcriptase PCR or RT-PCR, and real-time quantitative PCR, and also DNA sequencing. Let's look into the different methods that are available. Next slide, please. Okay, so this is an example of FISH. All right. FISH is a molecular cytogenetic technique. So it identifies a chromosome abnormalities using molecular technology. That's why it's called molecular cytogenetic. All right. The difference here is that it uses fluorescent probes. These probes hybridize only to complementary sequences, and the resulting signals are examined again under a fluorescent microscope, okay, as this picture shows. Next slide, please. The principle of FISH here is that the probe DNA with a specific sequence is labeled with fluoroprobes, which causes fluorescence, okay? So the probe, as shown here, light blue with red label, and target are denatured. During this denaturing process, usually we use heat. After denaturation, the target is then allowed to re-anneal, where the temperature is lowered, okay, and it hybridizes. And after we wash away the non-specific or excess probe, the cells are counterstained to show the chromatin location. And we observe the fluorescence under fluorescence microscope to determine the location of the probe signals as seen here. Next slide, please. Okay, there are different types of FISH probe. All right, for example, we have dual color, dual fusion probes, or break apart probes. And then we also have centromeric probes, and also use of deletion or duplication probes. So these are the different probes that are available. Okay, next slide, please. So this is an example of FISH interface. So in FISH, the cells are in interface as opposed to the conventional karyotyping where the cells are in metaphase. So we see here the BCR able translocation. Okay, here we can clearly see that there are two signals for each color in the normal cell. The red signal is for chromosome 9, all right, and then the green signal is chromosome 22, and we can see here that there are two red-green fuse signals indicating the fusion of ABL and BCR genes, all right. So this is using a probe that is located on the chromosome, all right. Next slide, please. Okay, the next the slide before that. Okay, this slide. All right. So besides which, we also have array CGH, which can be utilized in the in patients suspected to have genetic abnormalities. All right. So what is array CGH? It's a technique that was developed for high resolution genome-wide screening of segmental genomic copy number variations, or CNVs. Array CGH has a high resolution as compared to FISH and conventional karyotyping. So array CGH, its clinical uses is for detection of genetic abnormalities in leukemia and lymphoma. It's still research-based and not ready for standard use yet. It's also widely used in the diagnosis of genetic abnormalities in children, all right? Children with developmental delay, for example, intellectual disability, or children having autism spectrum disorder, all right? In this technique, in this process, in this method, we use equal amounts of labeled genomic DNA from a test, from a sample, and a reference sample. They are co-hybridized to an array containing DNA targets. Array CGH, even though the resolution is high, but it has its limitation, is unable to detect balanced translocations. Okay, the next slide will show in detail, all right? So step 1 is the patient DNA and control DNA. They are together, and then step 2, they are hybridized, and step 4, they are hybridized, and then step 5 and 6 is generated in the computer software, where the plot the plot will show the analysis of the data, and this has to be done by using a computer software. So step 5 is the microscanner as well, all right? You will use the microscanner to measure the fluorescent signals, all right? So step 1 to 3 is the patient and the control, which are labeled with fluorescent dyes and applied to the microarray, all right? And then step 4, patient and control compete to attach or hybridize to the microarray. Step 5, the microarray scanner measures the fluorescent signals. And last, but the computer software analysis will generate the data and generate a plot. All right, next slide, please. Okay, so this is again to show the profile. You can see either duplication or deletion of the particular area that we are looking at. Next slide, please. Next slide. Okay, so this shows the the results of the patients with abnormalities. Next slide, please. Okay, so this slide actually shows the comparison of each of the method that we have seen earlier. All right, so karyotyping, where we look at the whole genome, the genomic resolution is low, between 3, 4, and 5 megabase, right? As compared to FISH, the resolution is higher, which is 40 to 250 kilobase, looking also at the whole genome. And the chromosome microarray analysis, it's 30 kilobase, all right? Looking at the whole genome as well, all right? Next slide, please. Okay, so we proceed to the polymerase chain reaction. I'm sure everybody is very familiar with this method or this technique. It's a molecular technique used to amplify specific region of DNA sequences. It's the most commonly used molecular assay in the management of hematological malignancies. It has the main components. We need to have DNA templates. We need to have forward and reverse primers. We need to have an enzyme, which is Taq polymerase, nucleotides, and magnesium for PCR to proceed. Next slide, please. So this procedure involves amplification of 20 to 40 thermocycles, which comprise of denaturation, annealing, and elongation in each cycles. And we have to use some form of detection system, which is gel electrophoresis, to detect the amplicons or the PCR products. So the PCR fragments can be identified through size separation of the products on agarose gel electrophoresis. Next slide, please. So we have different types of PCR-based methods. One is the reverse transcriptase or RT-PCR. So this is the commonest type of PCR used in practice today. Here we use a reverse transcriptase, okay, to synthesize cDNA or complementary DNA from mRNA or messenger RNA. The final amplified product is assessed by agarose gel electrophoresis again. And the ability this RT-PCR is able to study the expression of genes in a qualitative manner. All right, next slide, please. So as opposed to reverse transcriptase or RT-PCR, we have real-time PCR, or sometimes we say RQ-PCR. So this is amplified DNA, which is tagged with non-specific dyes, which are fluorescent, that intercalate with double-stranded DNA. And then real-time PCR can quantify copy numbers of given DNA sequence in a sample, where the thermocycler contains sensors, okay, for measuring the fluorescence emitted from the fluorophore after it has been excited at a certain particular wavelength. So this generation of rate of fluorescence is measured, and real-time values of the DNA amplification can be achieved or can be provided from real-time PCR. Next slide, please. Okay, these are the commonly used systems in some of the labs here, and the commonly used specific gene detection utilizing real-time is for minimal residual disease monitoring in BCR-ABL and PML-RARA gene. Next slide, please. Okay, then we have Sanger sequencing. It was first introduced by a Nobel laureate, Sanger and his colleagues in 1977. They utilized chain termination method by fluorescent label dideoxy nucleotides, or ddNTPs by DNA polymerase during in vitro replication. This is a post-PCR analysis and a confirmation method for mutations. All right. So whenever we suspect the sample has new mutation or mutation not described elsewhere, we can use Sanger sequencing for confirmation of this mutation. Next slide, please. Okay, what happens during sequencing? There is again denaturation, where the DNA is divided into two strands, and then annealing, where there is primer binding. followed by elongation, where dNTPs are added to form new DNA, and also eventually there is chain termination where dNTPs are incorporated into the growing DNA strand, and thus stop the processes. Next slide, please. This slide shows the fundamentals of Sanger DNA sequencing, which I have explained and elaborated earlier. Next slide, please. Okay, this shows the fundamentals of Sanger sequencing again. So PCR to generate lots of target gene product. Okay, this is a repetition, and then we can detect the products using, capillary electrophoresis. Next slide, please. So then the sequence analysis software provides analyze sequence in text and the electropherogram, right? And the peak patterns reflect mutations or sequence changes. Okay, next slide, please. So this slide actually shows the sensitivity of the test, the different tests that we have mentioned earlier, okay, from morphology to PCR. So morphology, we are looking like at number of between 10 to 10 to the power of 2 cells. Cytogenetics, the sensitivity is higher. We are looking at the number of between 10 to the power of 2 and 10 to the power of 3 cells. For using immunophenotyping, looking at the immunological markers, the sensitivity increase here to up to 10 to the power of 4 cells. And of course, using PCR, we can detect the sensitivity is increased further by, we are able to detect at least 10 to the power of 5 to between 10 to the power of 6 cells. Okay, next slide, please. So these are the applications of molecular techniques and markers in haemato-oncology. So for diagnosis, for initial diagnosis, we can detect fusion genes or mutations, okay, using molecular techniques or genetic markers. That's for initial diagnosis. For treatment protocol, the value of molecular techniques, is for risk stratification and prognostication of the patient, and it is also useful and used to monitor response to therapy. Once the patient is given treatment, we can assess whether the patient is having MRD or minimal residual disease. Not only that, we can also monitor response to therapy when the patient undergo bone marrow transplant, where molecular techniques can be used to detect post-transplant chimerism analysis, and also monitor response to therapy in determination of lymphoid clonality in lymphoma or in Hodgkin's and non-Hodgkin's lymphoma. Next slide, please. Okay, so with the advent of molecular techniques in clinical genetics, we can see that there is very very great progress in the integration of these techniques in making diagnosis and also prognosis and therapeutics of the patient. This slide here shows the WHO classification for malignant, for hematopoietic and lymphoid tissues malignancies. All right. So before 2001, the classification utilized morphology and also immunocytochemistry. And the WHO classification has since been revised, where there is updated version, which is 2016, where in this classification, there is integration of morphology and genetics, all right? And with this information, it has generate a new diagnostic approaches to patients suspected to have hematological malignancies. And this in turn improve the prognosis, and we can predict the response of treatment to the patients by giving so-called targeted therapy. This is important towards the goal of precision medicine. Next slide, please. So this slide shows the different level of integration of genetic data into the clinical pathological classification of hematological malignancies. All right. For example, if we look at the genetic lesion pertaining to JAK2 mutation. So if we are able to detect whether JAK2 is present or not in a patient suspected to have MPN, so if the patient has this JAK2 mutation, this will help in the diagnosis of the patient. Okay, it can help you to delineate to make a diagnosis. All right. So in the prognosis, a patient having TP53 mutation or tumor suppressor gene TP53 mutation, which is present in several hematological malignancies, this will give a bad prognosis to the patient. All right. We can also utilize genetic lesions by predicting the progress, whether it's the patient having a good progress or not. Okay, examples of predictive factors are kinase activating lesions in BCR-ABL, like acute lymphoblastic leukemia. All right, and the other use for the other utilization of testing the genetic lesions is in disease-defining situations. For example, in patients with myelodysplastic syndrome with 5q deletion, so if they have just isolated 5q deletion in MDS, this will give a good prognosis in patients, especially with MDS. Okay, so here we term as disease-defining lesion. So these are the several advantages of knowing the genetic lesions of patients suspected to have malignancies. Next slide, please. Okay, so this is the WHO, looking at the WHO classification 2016, looking at further classification, taking into account the the genetic abnormalities. I think the next slide will be clearer. All right, so next slide. All right, so this is looking at AML with recurrent genetic abnormalities. Okay. So we can see, that knowing the abnormalities can define whether the patient has AML. For example, if a patient has AML with translocation 821, runs translocation, so regardless of the blast count, this patient is considered to have acute leukemia. All right. Next slide, please. So this is showing also molecular causes of AML and concurrent gene mutations. Next slide, please. Okay, and this is the recent AML risk stratification by genetics from European Leukemia Net. All right. So here. There is stratification by the genetics that are found in the patients. For example, a patient can be classified as having favorable, intermediate, or adverse prognosis. All right. So patient with favorable prognosis are patients here. We can see translocation 821 or inversion 16. All right. And patients can also be stratified into intermediate, for example, if they have mutated MP1, M1 or FLT3 mutation. Okay. And we can see here that, you know, there are a group of patients based on their genetic abnormality detected. They will be classified in adverse prognosis classification if they have translocation 6-9, for example. Right. So with this knowledge of genetic abnormalities, we can also re-stratify patients into whether they are having favorable, intermediate, or adverse prognosis. Next slide, please. So these are the list of 30 leukemia translocations that are available. Next slide, please. All right, so we have touched about, you know, the molecular techniques and its applications in malignancies such as leukemias. So before we go into the next section, which is looking at thalassemia and hemoglobinopathies, Dr. Yeti, do you all need a break or shall I continue?
Please you proceed.
Sorry?
Please you continue, Prof.
Continue, huh? All right. I thought you need a break. Okay. All right. So in thalassemia and hemoglobinopathies, molecular techniques have been applied. All right. And some of the techniques that we use here are polymerase chain reaction utilizing multiplex gap PCR for detection of gene deletion, and also multiplex arms PCR for detection of point mutation. Okay, there's also MLPA or multiplex ligation probe amplification. And also Sanger sequencing also can be used in the diagnosis, especially for confirmation of mutations, suspected mutations in the diagnosis of thalassemia and hemoglobinopathies. Next slide, please. Okay, for alpha thalassemia, the most common here are deletional mutations or deletional abnormalities. We have two categories. There's removal of only one of the two alpha gene and removal of both of the alpha globin genes, which can be identified by gap PCR. And the common mutation that we detect here is 3.7 and 4.2. All right, and also for the common, deletion of both alpha globin gene, we have SEA, MET, and Philippine, and 20.5. Okay, for the non-deletional forms, there are at least 20 mutations. The commonest being HB, hemoglobin constant spring, which is detected by ARMS PCR. Next slide, please. So this is an example of gap PCR for alpha thalassemia. All right, next slide, please. And then we have the beta thalassemia genotyping, which is mainly caused by mutations, point mutations, and the method for detection is using ARMS PCR. If the mutation is not known, we have to proceed to DNA sequencing analysis as well. Next slide, please. So this is an example of, ARMS PCR for beta thalassemia. Next slide, please. All right. So having said all that, we're talking about the hemoglobinopathies and thalassemia. Don't forget that we also have abnormality of the red blood cell membrane itself. All right. So we know that, the red cell membrane is a structure composed of lipid bilayer with proteins embedded vertically and horizontally. And this red blood cell membrane structure maintains the stability and normal discoid shape of the red blood cell. Okay, red blood. Sorry. Sorry. Tayati, did you say something?
Oh, masuk ya. Sorry, Prof. Narazah, do you want to break?
No, it's okay. I can go on.
Oh, okay.
No worries.
You continue. I'm sorry.
It's okay. I think we are almost 3/4 finished.
Oh.
Let's finish all first. Okay, can you go back to the previous slide? Previous slide, please. The slide before, please. The slide before. Okay. All right. So we need to have this membrane stability because red blood cell, as we know, the lifespan is 120 days, and then the size, the normal size is 7 to 8 microns. Sometimes during the course of its circulation, it has to transverse microvasculature, which is smaller than red blood cell size itself. So if the membrane is not stable, the cells can undergo hemolysis, all right? So this accounts for hemolysis, pathogenesis of hemolysis in patients with red blood cell membrane disorder. So the red cell membrane is important to preserve the red cell deformability, and the red cell membrane also has its antigens, which give its characteristics. All right. Next slide, please. And it also serve as selective permeability. All right. So in Malaysia, we have a high incidence of Southeast Asia ovalocytosis, which give the characteristic cells, red cells, to be ovalocytes. Not only they are ovalocytes, but they are big on the peripheral blood film. So we call them macro-ovalocytosis. How do we diagnose this? On the peripheral blood film, we have to have at least 25% of cells to be ovalocytes, but sometimes it can be complicated because sometimes in thalassemia, we also can have cells that look like ovalocytes. So the only definitive diagnosis is we have to resort to PCR-based assay. All right. So this is just a simple step. PCR, just one step analysis. The picture on the right side shows the results, gel electrophoresis results of molecular analysis to confirm patients which are heterozygous for 27 base pair deletion of this AE1 gene, or the Southeast Asian protein or Bantry protein gene. All right. And patients that are are heterozygous, you can see here that they have two bands here, all right, in lane 4, lane 6, and lane 7. All right, next slide, please. So that's the one of the application of molecular techniques, in delineating a patient, whether the patient have Southeast Asian ovalocytosis, and exclude from other causes of ovalocytosis, for example, thalassemia. All right. So this is this slide shows actually the application of NGS for detection of multiple mutations in the G6PD gene. So this is like a case report here. So I think we are very familiar with glucose 6-phosphate dehydrogenase deficiency, right? Is the commonest enzymopathy affecting the tropical and subtropical areas globally, right? And the disease is due to basically generally a single point mutation, or just a single base pair mutation. At least there are 250 mutations that are described. So once the G6PD gene is mutated, it gives the product is proteins with different levels of enzyme activity. Since this is X-linked, it generally affects the boys or the men, but ladies or women also can be affected. where they can either be heterozygous and homozygous, and sometimes they show symptoms. All right. So how the heterozygous women are affected? They show variations in the enzyme activity because of the DNA methylation and preferential inactivation of the G6PD wild type. Okay, next slide, please. So as mentioned here earlier, there are many mutations affecting the G6PD gene, and most are single missense mutation. They can also have multiple missense mutations, which are few, and sometimes they can have deletion or single nonsense mutation. All right. So if you look at the picture here, patients can have multiple mutation. That means more than one at one time. This happen in areas where especially G6PD deficiency is common. All right. So patients with multiple missense mutations have been described in the literature. Okay, next slide, please. Okay, in the diagnosis of G6PD deficiency, even though this disease is not, not, not fatal, not dangerous, not acute like malignancies, okay, but it can pose public health problem. All right. So in the detection and the molecular characterization, it can be very challenging. All right. Usually we have only biochemical analysis to check whether the patient has G6PD enzyme activity affected or not, and usually this is the method that are commonly used, which is using biochemical analysis. But there are limitations. All right. So in places where molecular analysis is available, patients may have the chance to undergo, or they can be tested using PCR to check for suspected mutations. But this usually occurs during hemolytic crisis. All right. So during hemolytic crisis as well, the patients may show nearly normal activity. So this is why molecular analysis is used. All right. So sometimes, we have to resort to Sanger sequencing, if we manage to find mutations that are not already described. All right. And then during the process of molecular detection of, the gene involved in G6PD deficiency, the patient has to undergo a lot of testing, which can be costly and time consuming. All right, next slide, please. Okay, so I will illustrate the utilization of NGS in a patient that we recently have. All right, I think you need to just enter, click. Please click enter. Okay, this is illustrated in a case which happened to one of our patient. She is a 30-year-old Chinese lady who came to see the physician when she was 28 weeks pregnant. She has had anemia since the age of 10. Enter. Okay, she was investigated thoroughly, extensively, for her anemia. And the only findings that were present at that time was the reticulocytes was 2.7, slightly raised, and MCV is high when she came to see the doctor during this time. The other positive history is she has the father has mild anemia but the hemoglobin was like 12.2 and MCV was 96. All right. So this patient was extensively investigated. So because because of that, she was recommended and to do NGS, and then the test was outsourced. All right. And the results came back as, sequence analysis showed the patient has 2 heterozygous missense mutations in position 1388, which changed from G to A. This changed the amino acid from arginine to histine, and she has another mutation 1376 G to T, which changed arginine to leucine. So these are pathogenic mutations. All right. Next slide, please. All right. So this finding showed that the patient has more than 1 mutation, and we know that these 2 mutations are disease-causing variants. All right, and the patient seems to have a compound heterozygosity of the identified variant. That's how it explains the patient's disease. And with NGS, the cause of chronic anemia is in this patient has been identified. All right. This case shows the application, illustrates the application of NGS, which is highly reliable and useful in G6PD diagnosis. Next slide, please. Okay, so molecular techniques also has been used for gene studies in lymphoproliferative disease in T and B cell rearrangements. Okay, we know that there is a significant variety of rearrangements of immunoglobulin and T cell receptor locus during the process of lymphocyte maturation. So any clonal rearrangement, sometimes which can be induced by viral infection, for example, can cause a hematological disorder. So this clonality can be detected in the peripheral blood of paraffin fixed or paraffin embedded blocks via PCR technology. So here we can see that molecular techniques can be used in the diagnosis of lymphoproliferative disorders. Next slide, please. So again, cryptic translocation prognostic significance, as we mentioned just now. Okay, the European leukemia NEC risk stratification by genetics. We have talked quite detail about these cryptic mutations and how the use of the information gathered here, we can stratify the patient, whether the patient can have favorable, intermediate, or adverse prognosis. All right. So here again illustrates the importance of molecular techniques in stratification of patients. Next slide, please. Also in MRD, minimal residual disease monitoring. All right. MRD can be detected and quantified in the peripheral blood or the bone marrow. And following MRD, is it helpful to organize risk groups and to monitor disease kinetics in refractory or progression case. All right. So with RT-PCR, we can detect at a level of 10 to the 4 or 10 to the 5 residual cell disease. Okay, or 1 leukemic cell in 10,000 to 100,000 healthy cells. All right. Next slide, please. So besides diagnosis, risk stratification, MRD, molecular techniques also can be applied for patients undergoing stem cell transplantation. All right, we know that donors must be selected and must be ideally matched for all these leukocyte antigens. All right, so the donor search utilizing, can utilize serological tests or genomic testing or NGS-based methods, which have been refined, but which guarantee high resolution results. Okay, so this is one of the other application of utilizing molecular techniques in stem cell transplantation. Next slide, please. Okay, so after transplantation, we also can utilize molecular techniques in patients having post-allogenic chimerism control to check whether, there is, the patient has accepted the donor's cells, all right? So a measure of the number of donor and recipient cells in the host following stem cell transplantation, we can predict whether, there is disease relapse or graft rejection or graft versus host disease. And the methods here, can be either FISH or, PCR-based methods, right? Next slide, please. Okay, and the next slide is, to show, I think this is the latest as of last year, 2021, September last year. These are the institutions in Malaysia that offer cytogenetic services, okay? We have in the Klang Valley, where most of the cytogenetic services are concentrated. For example, IMR, HKL, Hospital Ampang, UKM, some private hospitals such as SJMC, Prince Court, and PMC, all right? And in the north, in Penang, we have AMDI and also Penang Hospital, and also we have in the northeastern coast of Malaysia, which is offered by USM. Next slide, please. So these are some of the tests that are offered: leukemia translocation study, AML mutation study, CML mutation study, and chimerism study. Next slide, please. The red blood cell molecular hematology tests include, of course, thalassemia, hemoglobinopathies, red cell membrane disorders, and G6PD deficiency. Next slide, please. So this is a slide shared by Dr. Yuslina from IMR to show the different leukemia translocations which were detected in IMR, which comprise, 45%, the big group, which is translocation 922. And you can see some other translocations detected here, for example, PML, RARA, and also RANS1. Next slide, please. Okay, so this is acute myeloid leukemia mutation studies as done in IMR, all right? They use for diagnosis and prediction of response to therapy, and then also for prognosis of the patient using molecular mutations. This test has been offered since 2013. Next slide, please. This is the acute myeloid leukemia mutation studies in IMR, all right? They test for FLT3, NPM1, c-KIT, SEPA, and this test also has been offered since 2013. Next slide, please. Okay, besides AML mutation studies, IMR also offered chronic myeloid leukemia mutation study, all right? So the tests offered in IMR are PCR-able kinase domain mutations, all right? So with the discovery of imatinib in 2001, targeted therapy, which is TKI or tyrosine kinase inhibitor therapy, has resulted in its use as a frontline therapy. But we know that the long-term use of TKI occasionally results in emergence of therapy resistance, all right? So here we can identify patients who develop resistance to imatinib, okay? So these tests are offered in IMR. Next slide, please. Okay, so I think my next half an hour will be, or less than that, will be talking about future techniques that may be applicable. In fact, some are already being used in either diagnosis or prognosis or therapeutic management of patients with hematological malignancies. Next slide. Okay, next generation sequencing I've mentioned just now. As we know, NGS is we can detect parallel massive sequencing of genes at one time. So this is the application of high throughput, massively parallel sequencing technologies. There are different types of NGS, can be whole genome sequencing. Whole genome sequencing provides a comprehensive review of the entire human genome. Okay, and it covers both the coding and non-coding regions. The other one is whole genome, whole exome sequencing. It analyzes the protein coding regions, only the protein coding regions of the genome. Okay, and sequencing is restricted to exonic regions of the genome where pathogenic mutations are more common. And this is the more common sequencing NGS method that we use, whole genome sequencing. And the other one that is commonly used also targeted sequencing. This allows for the selective enrichment of particular genes or regions of interest. All right. And we use diagnostic lab for the evaluation of malignancies where the spectrum of mutation is well established and discovery work is not required. Okay, so targeted and whole exome sequencing are the most common techniques of types of NGS. Next slide, please. Okay, so this picture shows the whole genome and whole exome sequencing. All right, it shows the different exome size that can be detected by the different types of sequencing. Next slide, please. Okay, this is a picture to show a targeted sequencing. As we mentioned just now, it allows for selective enrichment of particular genes or regions of interest and is used in the diagnostic lab. Next slide, please. Okay, the other imaging technology that is worthwhile for us to look at is liquid biopsy. I'm sure you are quite familiar with the word liquid biopsy. So what is liquid biopsy? Next slide. Okay, in the era of precision medicine, liquid biopsy is increasingly being studied as a potential tool for refining our diagnosis and also for disease monitoring. The term liquid biopsy means that we assess tumor through blood sampling. All right, so we don't have to use, there is no need for invasive tissue biopsy. Next slide, please. How can we assess only through blood sampling? All right, in this liquid biopsy. So the fundamental here is that as the cells within the body die, they are broken down. Okay, and they are broken down into components of the cells, which include small DNA fragments, which measure about 160 base pairs, and these are known as CT, circulating tumor DNA. They are released into the bloodstream. All right, so they can be isolated by spinning down a blood sample and extracting it from the plasma. Okay, so in the presence of cancer cells, a small fraction of this DNA stems from the tumor, right? So this is called circulating tumor DNA or ctDNA. All right, next slide, please. So what is the current and potential application of liquid biopsy? One is we are able to establish the molecular fingerprint of the tumor at diagnosis. Okay, especially when the tissue is insufficient or cannot be sampled. Also, when the patient is too ill for biopsy, this is one of the advantages. and also at the time of progression to identify resistant mutations, because sometimes at this stage patient also may be too ill for biopsy. We also can confirm a targeted therapy and mechanism of drug resistance, also real-time monitoring of the disease, okay, for example, MRD. Also, application is for early detection of metastasis, all right? And at the same time, provide prognostic information. And it also can complement tumor molecule profiling before and at the same time or after the tumor biopsy, okay? Next slide, please. Okay, I think this is the last potential application of molecular techniques. I think you're also very familiar about CAR T cells or chimeric antigen receptors. So in this era of precision medicine, CAR T cells, one of the mode of treatment of patients with cancer, all right? So what is this CAR T cells or chimeric antigen receptor T cells? So they are proteins engineered to give T cells the new ability to target a specific protein. And the receptors are chimeric. Chimeric meaning that they combine both antigen binding and T cell activating functions. So this is combined into a single receptor. Okay, and the first approved treatment using CAR T cells, which target CD19, is already in use for B-cell derived cancers, for example, ALL and DLBCL, or diffuse large B-cell lymphoma. So we need to utilize molecular techniques if we are going to stratify patients into receiving CAR T cells treatment, and the tests that are beneficial or essential here are karyotyping, SNP and oligonucleotide microarrays, and NGS technologies as well, which can detect genomic structural variations. Next slide, please. So this table actually summarizes the clinical applications of molecular technology in hematology, which we have discussed in detail earlier. Okay, they are used for to detect inherited mutations, for example, thalassemia, also in gene studies in lymphoproliferative disease, to investigate for T and B cell rearrangements, also identification of cryptic translocation, especially for prognostic significance. Okay, also detect mutations in AML and lymphoid leukemia. Also for MRD, we also have seen its advantage usage in applications to stem cell transplant, also post stem cell transplant to check for chimerism, and also for CAR T cells, chimeric antigen receptor T cells treatment. Next slide, please. Okay, so in conclusion, all the molecular technologies that we have taught and described earlier give you an stems from the understanding of genetics, all right? And this in turn also has advantage to give us new insights to hematological diseases with respect to diagnosis, prognosis, and also therapeutic implications. So regarding diagnosis, these methods increase the precision, all right, and the speed of the results, and screening can be more easily performed. In therapies, targeted and immunomodulatory therapies such as CAR T cells guarantee better results with less hematological toxicities because they are targeted, right? They are biologically targeted. So advances in molecular genetics will continue to generate new diagnostic approaches, improve prognosis and predictive models, and also enable us towards targeted therapy for precision medicine. Next slide, please. Okay, so this summarize again the milestones of how the diagnostic tools has evolved in the laboratory. This is, actually a repetition of how we started earlier from morphology to NGS. Next slide. Okay, so with that, thank you very much for your attention. Here, I would like to say thank you, especially to Dr. Yus from IMR, for Yuslina IMR, for sharing the slides. Without which the presentation would not have been possible without her sharing of the slides with me. And then also thank you to Dr. Fitri. I hope Dr. Fitri is here, for introducing me to colleagues in Airlangga University. I first met Dr. Fitri in Bandung. She was the one that initiated our collaboration. Also to colleagues, Dr. Yeti and Dr. Pushpa, all right, and Dr. T. And if I forget to mention all the names, I'm very sorry, but from the bottom of my heart, I say thank you to everybody for making these efforts possible. And last but not least, to the management of ESF. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Prof. Narazah, also thank you to Dr. Thea and team as organizing committee that this event can be conducted today. Yeah. Dr. Thea, Dr. Sul, Dr. Pinar, yeah, Dr. Rifa. Thank you. Yeah. It's all your presentation, Prof, and this is a very complete, very clear, and very interesting lecture. I think, yeah, it is great and wonderful. And now we come to the Q&A, Prof. Question and answer. Do you want to break for several minutes, maybe for drinking or other, Prof?
It's okay. No? It's all right. It's okay, we can continue.
We can continue? Okay.
Yeah.
Yeah. In the chat room, we have several questions, Prof, and I will read the question. Yeah. The first question is come from Dr. Yuliana Dar Indrasari. Hopefully you still remember to Dr. Sari, Prof. Yeah. Assalamualaikum, Prof. Is it possible for detection of early relapse or minimal residual disease in acute leukemia with FISH method instead of immunophenotyping?
Can I look at the question, because I'm unable to view the question?
Bisa. Yes, I asked to my staff first.
Yeah, because I think my computer doesn't. Okay, I'm able to see the question.
Okay.
So, all right. From Dr. Yulia, okay. Is it possible to detect early minimal residual? Yes, instead of immunophenotyping. All right. Okay, I can't answer that, because what I'm very familiar is immunophenotyping and also doing PCR. But for fish, it depends on whether there is availability of the probes that is in the market or not. Okay, because as you know, for fish, there are we have to use designated probes to detect any aberration in the chromosome. All right. So I don't have experience using fish in to detect MRD in acute leukemia, but I know that we can use immunophenotyping, and also I'm very familiar as well with using PCR-based technologies to detect MRD. And the commonly used methods, as I have depicted in the earlier lecture, is doing real-time PCR, where we can quantitate. Okay, so I can't really share my experience using fish.
Okay, yeah. Thank you, Prof. Narazah. And you are prefer to test with the RT-PCR, yeah, rather than immunophenotyping to detect the MRD, Prof.
Yes.
Okay, yeah.
Yes.
Okay. Thank you. And the second question, Prof.
Yeah. Good afternoon.
Yeah. About the diagnosis of thalassemia and hemoglobinopathy, we use electrophoresis hemoglobin. This is from my student. And do we need a further examination like PCR? What kind of result can we expect from PCR? Yeah. Okay.
All right. Yeah. So for Miftah, Miftah Hasanah, thank you for the question. So traditionally, we use protein electrophoresis, such as HB electrophoresis, when we suspect patient to have thalassemia or hemoglobinopathy. But proteins, as you know, there are limitations, like in any other assays, for example, right? So of course, HB electrophoresis is our frontline investigation. But in cases whereby we cannot detect it through HB electrophoresis, for example, alpha thalassemia is not easy. Okay, it's not possible to detect by using the normal conventional methods. So here we use PCR based methods, like what I have shown earlier. Okay. So if, for example, in other complex diseases whereby the patient have alpha at the same time beta thalassemia, we also use PCR-based methods. Okay.
How about your experience, Prof, so far about the simple test for detecting the alpha thalassemia by watching in the microscope? We can look the golf appearance because it is a more specific test, yeah, and simple test.
Okay. In practice, what we do here is, of course, if we have the opportunity to look at the globin gene, which we don't have, because it's quite difficult to confirm the diagnosis of alpha thalassemia with conventional methods, right, Dr. Yeti? So sometimes, the patient may have just abnormalities in the counts, but the hemoglobin may be normal, and then sometimes they have just mild microcytic hypochromic anemia, for example, and then we have ruled out iron deficiency. So in our practice here, since we have the PCR-based technologies, we resort to doing to check for the mutation, the common mutations that we normally found in the situation. Sometimes we also call the family members as well to screen for thalassemia using FPT, the smear and the counts, and also looking at the other, family studies is also important as well if you don't have the liberty to use PCR-based methods.
Okay, so the PCR test is routinely examined for the patient of thalassemia in Malaysia.
Yeah, thalassemia in Malaysia.
routinely examine for the PCR molecular, Prof?
Yeah, I think
Okay.
yeah, for alpha and beta thalassemia is quite quite
Quite frequent.
generally used lah, quite commonly used lah. Okay, I think this is some of the tests that uh, generally quite commonly found in many hospitals or centers. I think thalassemia and hemoglobin because it affects a lot of our population, so the government has decided to give to support the testing for thalassemia as well, the molecular testing. All right.
Yeah, the further question is how to what we expect from PCR in order to diagnose thalassemia or hemoglobinopathy. Is there any difference from the PCR result from Prof for the thalassemia and hemoglobinopathy?
Sorry, can you repeat the question?
Yeah, the question that is still not answered, Prof. We expect from PCR, what kind of result that we expect from PCR in order to diagnose thalassemia or hemoglobinopathy? So it is any differentiation result from PCR between the thalassemia and hemoglobinopathy, Prof? What is the common?
Okay. Again, I'm talking from what we commonly find in Malaysia lah. For hemoglobinopathy, the common hemoglobinopathy that we have here, I don't know whether it's the same over there or not. We have hemoglobinopathy A.
Same. It is similar with Indonesia. Yeah.
Yeah. So for hemoglobinopathy E, we can directly make a diagnosis from HB electrophoresis, right, Dr. Yati?
Yeah.
All right. But if we cannot, from HB electrophoresis, we cannot, still we cannot identify. We will, go for the, suspected beta-tel, the common beta-tel mutations, using all the techniques that were described just now. And I think basically the limitation is that we go for the hotspots, the commonly found mutations that we have. All right. And this is usually what we find. All right. So let's say if the results do not is not compatible, we cannot find with the controls that we commonly have the mutations in our population, and we still suspect the patient has a mutation which can be a new mutation, we can resort to direct sequencing, okay, as the confirmative diagnosis. All right.
Yeah.
So we have the message here is that we have to look in general picture from the clinical information, clinical information, for example, the region where the patient comes from, right, and then the family history, and then their manifestation also from the full blood count as well, HB electrophoresis, HB analysis, and then you need to exclude iron deficient as well, which can be concurrent with thalassemia as well, right? So you need to exclude iron deficiency as well, and if you suspect patient to have concurrent IDA, we have to treat iron deficiency first. I think this is a common management as well. We give iron treatment to the patient for at least 3 months, right? See whether the iron is corrected or not. Only then we can do HB analysis, and the definition, the diagnosis can be more clear cut. All right. So in the message in general context here is that, we have to look at overall picture of the patient. All right. So only in difficult cases, only then you resort to PCR based technologies. But you cannot disregard the basic investigations and then the clinical information. Okay. I don't know whether I've answered the question, what kind of result that we expect from PCR.
Yes, I think it has been clear for your answer, Prof. But
Yeah.
we proceed to the next question, Prof.
Yeah.
The next question come from the Dr. Titi. Dr. Titi is our student also. My question, Prof. Narazah, as we know that the prevalence of acute lymphoblastic leukemia often occur in children age compared to adult. What is the cause? Is there any molecular detection of malignancy, hematology? Let me see. For prediction of infant during pregnancy.
Okay.
Yeah.
All right. Thank you, Dikas. Interesting question. Interesting question. At the moment, we still don't know, the causes of leukemia and also other cancers, whether it's solid cancers or blood cancers. Okay, we can only predict some of the causal risk factors, for example, viral infection, for example, EBV, Epstein-Barr EBV viral infection in lymphomas, for example, right, or Burkitt's lymphoma. We can just associate, but we cannot safely say that, a particular cancer is caused by a particular pathogenic factor, or we cannot identify the exact cause of any cancers, whether solid or blood cancers. So for patients with ALL, it's difficult to predict whether the patient is going to have in utero or not. All right. So that's why the answer to that is there is no molecular detection of malignancy hematology for prediction of infant during pregnancy. I think the answer is still no for that to detect malignancy during pregnancy. Okay, but of course we know certain conditions that can affect fetus when they are still in utero, for example, hydrops fetalis. Right.
Yeah. Okay. Yeah. Thank you, from Narazah. And it is also the invasive, very invasive. Yeah, I think to check the, maybe we take some amnion, from the pregnant woman. It is very dangerous. Okay, so we proceed to the next question, Prof. This is come from my student. Assalamualaikum, Prof. I would like to ask question. My question is for detecting MRT. We could use PCR for detecting the genetic mutation or abnormalities. Could we also use same method for initial detection? Maybe for PCR, yeah? Or should we use more advanced methods such as sequencing to assess initial detection? Thank you.
Okay. Assalamualaikum Umi Ilmi, ya. Alright. Okay. Okay. So can we use the same method for detection? Yeah, the answer is yes and no. All right. So PCR based can be used if we want to detect, especially for cryptic mutations. For example, the mutations that we have discussed just now, which confer risk stratification for patients, whether the patients have favorable or non-favorable prognosis. All right. So the reason why real-time PCR is used to detect MRT is because we can quantify and then we can monitor the subsequent progress of the patient. All right. So could we use the same method for initial detection? Yes. All right. We can use PCR-based techniques if we suspect the patient to have cryptic mutations or other mutations which can affect the prognosis or affect the prognosis, or we want to predict the patient will respond or not to the treatment. Okay. And then sequencing, as I said, is basically, if we want to confirm, for example, new mutations, or and then if we want to detect multiple mutations at one time, okay, we can use NGS. But I didn't mention about the price, it still can be expensive. NGS, for example, the patient that I showed you illustrate just now, the patient with G6PD deficiency, the patient has to pocket use the patient's own money for for outsourcing of NGS. So again, price is the factor here. I hope that answers the question, Dr. Yeti.
From the Dr. Yesi, my junior staff, would you like to explain more about cryptic translocation and NGS?
Okay, I think I mentioned several times about this cryptic translocation. So this cryptic translocation actually cannot be detected by our conventional cytogenetics. It may be due to the reason that it's, it affects the DNA definitely, but you cannot detect when you use conventional karyotyping, right? Because conventional karyotyping, the resolution is low, between 3 to 5, and then the way to go about is to use PCR based methods. All right. So PCR based methods, whether it's one step PCR where you are detecting only one particular mutation, or if you want to detect multiple mutations, of course the ideal way is to use NGS. But if, let's say, you are targeting just one particular mutation, you can use just targeted or just PCR to detect that particular mutation. But there is a limitation, because if the patient has other mutations in the gene, you are not able to detect the other mutations, because you are very specific. All right, you are just using the primers that are detecting that particular mutation. So the other option is to use NGS, where you can detect multiple mutations.
Yes, right, Prof. Is the NGS also available in the USM, Prof?
Unfortunately, no. We are still fighting to get it in my place, but in Kubankrian, yes, where Prof Rostin is located, they have just purchased. They are going to use for solid cancers, and they are focusing on research first. So for patients who can afford, they especially in private hospitals, they have the option of of outsourcing, but the patient has to pay themselves.
Okay, yeah. So in our institution also still not yet have, the NGS. Okay. The next question come from my staff lecturer, Dr. Betty Agustina. I want to ask, how could we detect that there is graft rejection from molecular testing, Prof?
Okay.
It is focused on the hematology malignant graft or transplant, or yeah, maybe other general, yeah.
General. How do we
or
detect
hematology transplant maybe?
Let me try to understand the question. How do we detect graft rejection from molecular testing? I think I've covered that in the one of the notes just now, right? One in the slides.
Pardon me, Prof?
If we refer to the slide that I presented just now, whether there's graft rejection or not from molecular testing, I think I have. Let me check my slides. Can't remember now. So this is, you want to see whether there is, the patient has taken the graft donated or not, right, from molecular testing.
Yeah.
I think that's the question. Is it like that, the question?
Dr. Betty, masih adakah Dr. Betty? Mungkin bisa menjelaskan langsung.
Dijelaskan?
Ya.
Yeah. Sorry, Prof.
Yeah.
We do transplant, some transplant in our hospital, like kidney transplant and stem cell transplant. But we don't know how we know that there is rejection from the transplantation. How could we know or we detect that there is rejection from our transplantation?
Okay. All right. As far as I know, if let's say you do not want to apply PCR-based techniques, if there is, if the transplant is from different sex, donor, right?
Yeah, for kidney transplant from related donor.
From a
related donor can be different sex or the same sex.
Yeah, kidney transplant. I think basically you can apply the same concept for hematopoietic stem cell transplant, because if the donor is from different sex, then you can look at the karyotyping, if you don't have facilities for PCR. For example, if the donor is from XY, from a male donor to XX female donor, you can do a buccal mucosa. You can check whether the patient has engrafted, because the sex chromosome will change. All right, but of course, I don't know whether you can apply. Maybe you can apply the same technique for kidney donor, but I don't know how fast, how fast the kidney donor, the kidney transplant patient will accept the donor. Maybe it's not the same time frame as hematopoietic stem cell engraftment. All right. So as I mentioned earlier in the lecture, you can again use PCR based techniques for hematopoietic stem cell transplant, or again NGS lah. And then if you don't have all that, again, as I mentioned, use karyotyping if the donor is a sex mismatched donor. For kidney, I'm sorry, I can't really satisfy. I don't really have information. No experience, Dr.
Betty.
Dr. Betty.
Yeah, no problem, Prof. It's okay.
Thank
I
you,
think,
Prof.
yeah. Can we continue to the next question, Prof. Narazah? Still 2 questions in the chat room.
Yeah.
Yeah. Okay.
1 question or 2 questions?
2 questions, I think.
2 questions. Okay.
Dr. Wuli. Assalamualaikum, Prof. For organ transplantation, if we don't have NGS, it is please share if we use test panel reactive antibody. Yeah, almost similar with the question of Dr. Betty.
Yeah, organ transplantation. I don't understand what is test panel reactive antibody. Is it antibodies towards red blood cell or what?
Yeah, maybe we continue the next question. Yeah, Prof.
Okay. Yeah.
From Dr. Fitri. Yeah, Dr. Fitri. Please, Dr. Fitri, it is honor for you if you will deliver your question directly to Prof. Narazah because Prof. Narazah miss you.
Yeah.
Please you show your face, please. Dr. Fitri.
Iya, dokter, mohon maaf saya di jalan, dok.
Oh.
Assalamualaikum, Prof. Narazah.
Yeah.
I'm
Waalaikumsalam.
very happy and it's our pride to have this expert lecture from Prof. Narazah, and we are very happy for this collaboration, Prof. Thank you very much. I have one question. Is there any commercial kit for in vitro diagnostic for diagnosis of alpha or beta thalassemia, Prof? Thank you, Prof. Narazah.
Okay, thank you, Dr. Fitri, for the question. Is there a
Good.
commercial IV in vitro, is it?
Yeah.
Okay. I am not familiar with the kits because we haven't purchased any such kit. But from my experience for prenatal diagnosis, all right, for prenatal diagnosis, we can have we can use again the known mutations that have been described and found in patients suspected to have alpha or beta thalassemia. All right. So if you were to ask me which company or which company actually have this commercial kit, I won't be able to provide because we don't really have prenatal diagnosis. Prenatal diagnosis is only found in only some certain hospitals in Malaysia, especially the private hospitals. And then the private hospitals, they have facilities to check for, to screen for thalassemia. So they will send to private labs as well for the, for this prenatal detection of molecular alpha or beta thalassemia. All right. So they are more free because, they are more free to choose which facilities, private facilities or private labs that they have. But for us, because we don't have prenatal diagnosis for alpha and molecular, we do only for, patients who come with clinical suspicion. So we don't really use the kit. Okay, we usually prepare ourselves. All right.
Yeah, usually we design the primer, by ourselves, Prof. And we order to the lab or referral lab to.
It's cheaper.
This is I give the opportunity to Dr. Woolley to deliver the answer, because we cannot understand what you mean. Please, Dr. Woolley. Are you still monitor, Dr. Woolley? Dr. Woolley.
Assalamualaikum, Prof.
Waalaikumsalam.
Excuse me, Prof. I mean that panel reactive antibody is usually used for detection of antibody of HLA. If for organ transplantation, if we don't have NGS, it's pleasure if we use panel reactive antibody to detect the antibody HLA. I mean that. Thank you.
You mean that if the patients develop antibodies to particular HLA antigen, so you can tag the antibodies, is it? You can check for that particular antibodies.
Yes, Prof.
Yeah, I have no experience actually, Dr. Mulis.
Yeah, but
thank you, Prof.
About the NGS for organ transplantation, it is right, Prof. Narazah.
Pardon?
NGS for checking the what is the
For
maybe monitor.
HLA antigens. Yeah.
Oh,
Yeah,
XLA.
that's the new method. But you know, before this we use serological testing, right? Yeah, and then sometimes we resort to just PCR based techniques, not NGS. So when I say NGS, is the the latest scenario. But of course, we most of the labs now still use serological methods plus PCR based techniques, bukan NGS lah.
Yeah.
Okay, NGS ni mahal.
Ya betul, Prof, betul.
Yeah.
Karena wool itu yang bikin mahal.
Betul.
Ya. Prof
Thank you,
Narazah,
Prof.
I think the Dr. Muli's questions is the last question for this moment. Yeah, I thanks very much to you, Prof Narazah, for the great lecture and the interactive discussion. Yeah, dan juga untuk semua partisipan dan peserta yang sudah berpartisipasi hadir di Zoom ya, dan telah memberikan pertanyaan yang interaktif sekali ya, yang menarik sekali. Saya ucapkan terima kasih. Semoga acara ini bermanfaat ya. Atau masih ada yang ingin bertanya lagi? Tidak ya, sudah too long enough. I think Prof Narazah has give the lecture and answer the question. Thank you very much, Prof Narazah. Maybe you have some message for us, Prof, before I close this event.
Okay, thank you again to the organizing committee, and then especially headed by Dr. Tia. Dr. Tia tak nampak mukanya.
Dr. Tia tak nampak mukanya. Please,
okay, ramai sangat kot.
Please give greeting to Prof Narazah, Dr. Tia. Dr. Tia is in the one room with me,
in
Prof
one
Narazah.
room. Okay,
Yeah.
okay. Alright. So, alright. Saya tak ada apalah nak cakap.
Ini Prof.
Just again to. Okay, Dr. Tia. She has always been very, very efficient in getting information from me lah. Saya tak boleh escape lah. Alright. So saya tak ada apa, cuma saya ucapkan ribuan terima kasih semualah kepada organizing committee dan juga semua para peserta lah. Saya tak pandai nak cakap bahasa Indonesia, tapi saya dengar Dr. Yeti cakap bapa-bapa, ibu-ibu dan juga students lah. Alright, insyaAllah we'll meet again, and if there's any question, maybe we can communicate by email, and I think we are going to meet again lah next week. And I hope kita doakan lah collaboration ni akan berjaya lah. We work hard towards that lah, Dr. Yeti.
Yeah. So the next lecture is about the molecular. What's your next lecture? I forget the title.
It's something similar.
Okay.
Something similar, particularly concentrating on AML, but I'm going to share some of our research findings lah.
Okay.
Maybe a bit boring.
Yeah, in 24, yeah. 24, yeah. Okay, insyaAllah.
InsyaAllah.
Okay, thank you, Prof Narazah, and dan Bapak Ibu semuanya. Kita akhiri acara ini dengan ucapan hamdalah ya, alhamdulillahirabbil alamin, sudah terselenggara dengan lancar dan baik ya, atas izin Allah, dan kita tutup ya, kita akhiri. We will close this meeting, Prof Narazah, this lecture, and thank you, and we can live together. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Prof Narazah silakan. Kalau mau live, saya mau live juga tanya. Mau sholat asar, di sini sudah asar, Prof. Baik, saya live ya, Prof Narazah.